Welcome back, friends. In this video tutorial we'll be talking about the mode of action of antibiotics which are interfering with the pathway of some important what you can say molecule cellular molecule production, molecules like precursor molecules, precursor for DNA, RNA, these molecules. Now inside the cell there are different metabolic molecules like precursor, as I have talked before. Now those molecules are required for production of nucleic acids. Now one of such precursor is what you know as usually folic acid or folate, right? So let me talk about it. So folate is one of the important ingredient or precursor molecule to produce nucleic acids. Now inside the cell the production of folate requires a pathway as a metabolic pathway. Now for that metabolic pathway to occur properly there are different enzymes to be function, right? Now due to any kind of antibiotic action, if those enzyme activity is getting altered, as a result the folate production will be decreased. Now if the folate production is decreased, then nucleic acid production will also decrease. Now especially what they produce this using this folate, what they produce is purine and pyrimidines. Those are the building blocks of nucleic acid, we all know. Now if there is a less activation or less production of nucleic acid contents, like less production of purines and pyrimidines, in those cases cell won't get enough amount of DNTPs, and as a result of not getting enough number of DNTPs the cell will not function properly, cellular machineries will be halted, right? So in this video we'll be talking about such different kind of those kind of antibiotics which are blocking those metabolic pathway. So metabolic pathway blockers, and among the metabolic pathway blockers we'll be talking about the mode of action obviously, and here the example is sulfonamides. Sulfonamides are the name of the group which is consisting of many different antibiotics, and one of the such antibiotics is sulfomethoxazole. Okay, sulfomethoxazole is an example. There are 2 or 3 different examples. I usually don't remember all the names. Sulfomethoxazole is a example of antibiotic of the group sulfonamides which will interfere. So let me talk. Interferes with purine or pyrimidine, especially for purine synthesis. Okay, synthesis. So it usually interferes with the purine or pyrimidine synthesis. Okay, so before understanding the mode of action by these sulfonamides, you need to understand the process, the cellular process or pathway of the purine synthesis. Now we don't need to know the whole pathway. We need to know the pathway until the production of folic acid or folate. Okay, so what is going on actually inside? We are having 2 important things. One is dihydrofolate. Dihydrofolate should be F. Dihydrofolate diphosphate. This is a precursor molecule. Along with this dihydrofolate diphosphate, what we also require? We require PABA or para-amino benzoic acid. Okay, so that means it is having an amino group at the para position of the benzene ring, right? So that's why you call it a para. It is having a structure slight like this. It's a para position amino acid like this or something like that. So it's called the para-amino benzoic acid if I'm not wrong. Anyways, so let's use the confusion. So PABA or para-amino benzoic acid, this is, and also dihydrofolate diphosphate. These 2 things are the precursor molecules to produce hydrofolate which is required. Now after combining these 2 things together, what they can produce is dihydropteric acid. Dihydropteric acid. Now in different books you may find different types of pathway. Some books elaborately discuss the pathway. I am here discussing very basic thing of the pathway. Now from this dihydropteric acid, what it can be further converted into dihydrofolate or dihydrofolic acid. Dihydrofolic acid. Okay, now from this dihyd, not folic, I think this should be F instead of pH. I'm not sure, but anyways, dihydrofolic acid. It should be F. Dihydrofolic acid. Now from dihydrofolic acid or dihydrofolate, it's the same thing. It can further convert it into tetrahydrofolic acid. Tetrahydrofolic acid or tetrahydrofolate. Right, so this is the basic scheme of the pathway. Dihydrofolate diphosphate along with para-aminobenzoic acid convert themselves into dihydropteric acid. From dihydropteric acid or dihydrofolate, it converts itself into dihydrofolate. From the dihydrofolate, it further converts itself into tetrahydrofolate or tetrahydrofolic acid. Okay, so this is the scheme of this whole process. Okay, now in this whole pathway there are enzymes, majorly two enzymes playing the vital role. One enzyme is acting here in this place, and that enzyme is called as dihydrofolate synthetase, and another enzyme is acting here at this last stage of conversion, and in this case the enzyme is called dihydrofolate reductase. These are the two enzymes, dihydrofolate synthetase and dihydrofolate reductase, which are playing important roles in two different stages of this whole pathway. Okay, now in this case of this kind of sulfonamides like sulfamethozazole, okay, now this kind of antibiotic, they usually interferes with this step. That means here this dihydrofolate synthetase is an enzyme, and these two things, PABA and dihydrofolate diphosphate, these are the substrates. Like these are the substrate, and this is the enzyme. Now what we know about the enzyme substrate reaction is that enzyme will act with substrate, and what it will produce? It produce enzyme substrate complex. Now from this enzyme substrate complex, it will produce enzyme plus product. Now we are interested about the products, right? And it is kind of reversible in this case. Enzyme substrate formation is a kind of reversible like that in most of the case. Okay, like that. Now this part is important. Now in this case, this sulfonamide antibiotics usually interferes with this enzyme substrate binding, right? How? Usually this sulfonamide antibiotics acts as a competitive inhibitor for this dihydrofolate synthetase enzyme. That means competitive inhibitors are those chemical molecules which interferes with the substrate for the binding with enzyme. That means usually this enzyme must bind with PABA and dihydrofolate diphosphate to convert them into dihydropteric acid, but instead this sulfonamide antibiotics, so if I write AB here, sulfonamide antibiotics, it will compete with these two substrates, PABA and dihydrofolate synthase, to bind with this enzyme, right? So if this antibiotic binds with the enzyme, if the antibiotic present in higher concentration than this PABA and dihydrofolate diphosphate inside cell, they will more rapidly bind with the enzyme, and all the enzyme will be filled with this antibiotic. So no single enzyme will be there to convert PABA and dihydrofolate diphosphate into dihydropteric acid. As a result, rest of the steps will not be occurring. As a result, there will be very few tetrahydrofolate, and as a result of that, there will be no purine or pyrimidine synthesis. And if there is less purine or pyrimidine synthesis inside a cell, there will be less nucleotide sequence. If there is less nucleotide, then in most of the processes, remember, DNA replication, transcription and translation, in all this case we require DNTPs, we require nucleotides for the joining, for the synthesis of the DNA or RNA. Now if there is less concentration or less amount of less amount of purine pyrimidines or less amount of nucleotides present there, the process of replication, the process of transcription will be halted, and that's what is done by the sulfonamide antibiotics. They are blocking the stage here. So if I draw here the antibiotic, this antibiotic is going to inhibit this stage, is going to inhibit the stage. As a result, no production of nucleotides. Finally they are going to produce nucleotides, but as a result of this blockage, there will be less nucleotide inside the cell. That's what is done by the presence of sulfonamides. An example is sulfomethoxazole. Okay, so this is a mechanism played by sulfonamides. It's completely different than other types, and you may find a difficult kind of pathway, but this is the basic kind of thing. There are certain other kinds of antibiotic present there which are going to interfere with this dihydrofolate reductase enzyme. Right now, here they're interfering with the dihydrofolate synthetase enzyme. Okay, anyways, so that's the process of action, and I hope that's helpful. Thank you.